Thank you very much. My name is Edgar Vallejo. I'm a professor in Monterrey Tech in Mexico, and I'm a collaborator in a research project with Professor Charles Taylor at UCLA. And this work was done, this is an ongoing work that has been done along with our students, both from UCLA and Mexico. So the outline of this talk will be, I'm going to explain the research subject and area, the experimental framework that we used, the problem that we are addressing, individual classification, some preliminary results, and conclusions. So we're studying right now Cassin's vireo. This is a small bird that is very common in western U.S. It ranges from Canada, British Columbia, western coastal U.S. states, and it migrates to Mexico during winter. And one of the main characteristics of this bird is that it sings constantly. So this is a map of sightings from eBird. So the song will be played right now. So the Cassin's vireo song consists of very short phrases that are spaced about 2 seconds apart. And they are, the phrases are, could be stereotyped. Here are the spectrograms. And basically, there have been some recordings that has been taken in the field. So the general experimental framework of the project has been, this project tries to use sensor arrays to understand bird communication, and we are actually collaborating in this part, the part that is related to species and individual recognition. And the data that has been collected for this study were located in the foothills of the Sierra Nevada, in the volcano area in California. The dates were from April 25th to June 18, 2013. The number of recordings were 56, and the duration varies a lot. It varies from 2 minutes to 2 hours. And the recordings were taken from 7 individuals. Each recording includes from 4 to over 1,000, over 1,500 phrases. So the phrases have been identified visually from Cavi songs using the Pratt software. So that was done in Chuck Taylor Laboratory, and they identified over 100 different phrases. So the songs were then represented as strings of phrases again. So this is a subset of the, the master list of phrases that consist of over 100 different phrases. These phrases, have been confirmed using machine learning, specifically support vector machines. And they found that the phrases were classified correctly, over 95% accuracy. So in the past, we were applying machine learning techniques for identifying this type of birds, the antbirds that are present in Mexican rainforest. So the song of this type of birds is pretty simple. So we were able to get very good results using different machine learning techniques. Specifically, we've been using classification trees. This is a supervised method, supervised learning method. We've been using also unsupervised learning methods like self-organizing maps. We use it also fuzzy logic, super vector machines, and hidden Markov models, of course, and getting good results with this type of birds that has a relatively simple song. Well, for Kabi, it's kind of different. You know, they have a very complex song structure. Let me show you. Our students are preparing a mathematical library for analyzing bird songs. The library is called Pájaro Loco. And, you know, they are checking, for example, the use of repertoires, the frequency of syllables, and they also modeling, you know, the networks, both the Markov networks and the centrality networks, and also the zip distributions here. So at this moment, it's just a visualization of modeling. It's a descriptive modeling, but we plan to incorporate some classification functionalities to this library. So, so you can see this is not the more complex Markov chain of the song, even though it is not, you know, it has some kind of complex structure. So the idea is that there was somebody that mentioned that when we have sequences like this that consist of strings of phrases, we can look at different features. I mean, we can look, for example, at acoustic features, both temporal and spectral features. And also, we can obtain the statistics of sequences. For example, we can count the frequencies of the different syllables. We can also construct graph-based models from the sequences. And this accounts for example, finite state automata, Markov chains, and hidden Markov models, for example, and language networks as well. We can also look at phrase dependencies. For example, we can look if the occurrence of one phrase in some way affects the occurrence of other phrases in the sequence. So this is, I think, this is a potentially useful method to look at the structure of songs, because dependencies have a direct mapping with Chomsky hierarchy. So we can see if these dependencies are how they are organized. If they cross, for example, they would belong to the context-sensitive languages. If they are nest, they will belong to the context-free hierarchy, for example, context-free class, for example. We can also, look at the motifs of the sequences, patterns that doesn't occur, consecutively, but it occurs, with some phrases interspaced. So we could also look at the grammar. This is a context-free grammar that could generate this sequence. The idea is that if you look at different aspects or attributes of the sequences, you'll be able to classify them using different mechanisms. For example, if you have a grammar, you'll be able to construct a parser automatically that looks at specifically look at grammatical features. And for example, if your biological question is related to assess the geographic variation of songs, then probably the dependencies would be the best feature to look at. if you're looking for a different problem, probably you have to take a look at different features. So I think that it's important to be able to analyze them. And fortunately, there are many, many techniques that could be used for this analysis. So our first question was to check if we are able to identify individuals just by looking at one phrase. I mean, the input of this classifier would be just one phrase, and the output will be the individual. So we use a collection of over 7,000 phrases that are described by the acoustic features of a song to classify 7 different individuals. And we use support vector machines for that, and also cross-validation. For this study, we use a popular machine learning suite, which is Weka. It's very easy to use. And we obtained these classification results. They are not really good compared with the ones that we had previously with the other birds. So we just classified correctly about 2/3 of the songs. You can check the confusion matrix and see, for example, that the individual UCT was not identified properly. It was confused with other individuals. But, mostly, the main phrases were classified correctly, but still we have a huge error. So it's not very accurate. We tried several different machine learning techniques, different attribute selection techniques as well. But this is the one that gave us the best results. So apparently we have to look, have a broader perspective of the sequence in order to be able to classify them correctly. So what we did is to construct the vectors of repertoires. The repertoires were described first by binary vectors. I mean, if the bird uses that phrase, it will have a 1. If they don't use it, it will have a 0. And also we construct the frequency vectors that have the percentage of use of that specific phrase. And we used the same individuals. And for this time, we used different, as a demonstration, we use different techniques, probabilistic, also classification trees, neural networks, and again, support vector machines. And this time we have much better classification, for example, with the binary vectors. These are the techniques. The one that, at least for these experiments, had the best accuracy was the multi-layer perceptrons, which is a neural network model. And close to that was the support vector machines. But when we use the frequencies, we got the best results with more or less the same results with support vector machines, and again the best results with neural networks. And there are some advantages and limitations with the machine learning methods, but just as a demonstration, we can see that classification trees could be very convenient because they yield a tree that you can use to infer classification rules. For example, here, if the verb has the AD phrase, then it should belong to a specific individual. But if it has other one, then you can create rules to classify them differently. So this is kind of nice because it gives you the reason why the model is classified in that way. And I mentioned, we're constructing a library that will be used in the Mathematica package. Then for the moment, it's able to show you some descriptive features of the sequences. And also, we are devising at this moment a method for classifying individuals based on sequence alignments, similar to the one that we use for BLAST, some molecular sequences. So the idea is that we will have the 55 sequences, and the idea is to compare these sequences against the consensus sequences of each individual. So you have to align first all of the sequences that belong to the same individual. We use a global alignment algorithm that is called Needleman-Gonch. And then once we have the alignment, we obtain the consensus sequence. And then finally, we use local alignment to compare the sequences against the prototype of each individual. So let me show you some graphs that show these ideas. The colors are not showing really well, but this is global sequence alignments. You can see the green ones are the coincidences or matches, and the pink ones are the mismatches, and we can also have gaps. But this is a convenient way to check for common subunits of the sum. So the idea is that once you align this sequence against all others, then you'll be able to create a profile or prototype sequence of that particular individual. Then you use local alignments to compare the sequence against the prototypes. So the idea is to, once you compare the new sequence against the prototypes, you can get percentage of similarity to that specific sequence. So we have obtained very good results. I mean, so far, all of the sequences belonging to specific individuals have obtained the best score to the prototype of that individual. We have also begun to look at the Markov chains of the songs. We just had a very few experiments so far, but we were able to classify over 90% using hidden Markov models with, and also Markov chains for classification. Finally, we're looking at this moment, this work is ongoing. We're looking at the collocations. This work is related to the dependencies detection. So the idea is that try to determine if the occurrence of two phrases create a dependency with each other. So collocations could be taken as a preliminary evidence that the one specific phrase is influencing the occurrence of the other. So basically, we have counted the frequencies of the collocations, and also we have obtained some scores. And the idea is that we can use also the collocation profile to classify the individuals based on that information. So that will look at dependencies at longer distances. I mean, they will not look specifically at adjacent phrases, but they will look at more separated phrases. So far, we can say that phrases seem to be insufficient for classifying individuals. I mean, phrase, it should be, probably it is too short. The information that is there is not sufficient for accurate classification. And repertoires allows, this confirms the observation of the field work that has been, I've been studying the field work that the repertoires are sufficient for classifying individuals. And sequence alignments seem promising. And in general, I think that there are many methods in bioinformatics that are used for sequence analysis that could be applied for some classifications. For instance, looking at specific motifs that could be probably functional units of songs. And there are many algorithms for doing that. The problem so far for adapting these algorithms is that our phrases are represented by two symbols, and generally the programs that make the alignments use one symbol per position. So that's the idea. So that's the conclusions. In the future, I think that we should be able to construct scoring matrices like the Blossom 50, the molecular sequence alignment use, and that would allow for better sequence alignment. I mean, if you have account for you can measure the similarity of different phrases, you'll be able to align it better. Also, hidden Markov models could be, could be combined with alignments that probably you could be able to identify specific units that are very important, are conserved in different sequences, vocal sequences. We could also like to explore assembled learning. I mean, to combine the results of different classifiers in order to make it more accurate. And also, we think that grammatical inference is necessary if we like to understand the meaning of songs. In general, I think that one specific, very important problem is try to see or to understand if the principle of compositionality is held in the animal vocalization. This principle says that the meaning of complex phrases could be inferred from the meaning of smaller phrases, of the constituent phrases. So the idea is that if we like to do that, we should be able to describe the language with using recursive mechanisms like grammars. And I think that there are many techniques for grammatical inference, but the computational problem is intractable. I mean, inferring a grammar from examples is not easy to do. Okay? So, but there are many data-based techniques that could be useful for trying to infer a grammar from this data. And also, we like to check what's the usefulness of these classifications in different contexts. I mean, probably for assessing or recognizing not just individuals, but for recognizing populations that has geographic variations, for example, among other social and geographical problems. So that's it.
Any questions before we move on?
No, I'm just speculating that probably, this feature could be useful for assessing these differences, but I'm just speculating about that. Yeah, that's correct. The sequence, the sequence alignment, the global one tries to align the sequence with the other, not with the prototype. First, you align all of the sequences of the same individual, and the problem is, you're right. When you have a very short sequence and the other one is a very long sequence, you can have many different alignments that have the same score. So that's useful. That's the reason that scoring matrices could make the alignments more accurate. Yeah, yeah, I agree. I mean, dynamic time warping is another technique that could be used. This part is just, like a proof of concept. I think that could be applied better for sequences that has mostly the same length. And it's just an idea, but I think that, it could be improved, definitely. I agree.
Just one to delve in discussion, the Needleman-Wunsch for short sequences essentially reduces to the edit distance because alignment doesn't come into play when you're talking about short distances. So I think it's still a kind of useful thing, but maybe we can discuss that later.
What kind of what? Sorry? I'm just using the edit distance right now. Yeah, just looking at mismatches or gaps. So as Eric said, is the edit distance. So how many operations do you need to take one sequence?
If anyone here knows a way for any system to say that the animal considers this change to be more significant than that change, then you can build a scoring matrix which is biologically relevant. But anyone says so, then I don't know how to do that.
I'm sorry, I couldn't
How
hear.
do you think you'll construct the scoring matrix?
It could be constructed from data. Like most of the alignment scoring matrices for proteins were constructed for many samples of data. So we can use distances in the spectrum, in the spectral space. I don't know, frequency space, I mean. But I'm not sure really at this moment would be the best way to do that.
Are there any other specific questions for this talk? Should we move on and open up a wide discussion?
Just a question. When you were building your consensus matrix, were you doing it based on just one set of training data, or did you go back and experiment, like for instance, at a equivalent of bootstrap, and
Yeah.
see whether or not the
Yeah,
standard
we do that. We basically use the leave one out cross-validation. So we constructed the prototype with n-1 and tested with the one.
much difference in your consensus matrix?
Not, yeah, sometimes, but the accuracy, was consistent, more or less. At least for this sample. This is very preliminary. Mm-hmm. Thank you.